with subsequent exposures, we actually have higher antibody affinities and titers that occur. So here, if we look at different times of immunization and antibody levels, looking at their concentrations, you can see that after the primary immunization, we have a spike in IgM antibody levels, and then we have an increase of IgG class switched molecules. However, after a secondary immunization, we have a higher titer of IgG, so class-switched molecules relative to IgM, and we really don't see an increase of too much IgM with subsequent infections, but we always see a spike in IgG or class-switched antibody levels. And then if we look at the affinity of these antibodies, IgM molecules, as they suggest, as they don't undergo very much somatic hypermutation, their affinity stays constant throughout multiple immunizations, but class-switched molecules will always increase in affinity after subsequent infections or immunizations. And what's really interesting is that you can actually see this property if you infect or immunize with different pathogens. So in this example, again, this is looking at the amount of antibodies, so antibody concentration within our host, across the time of different exposures via infection or vaccination or what have you. So here we have a primary immune response to initial exposure to pathogen or epitope A. And you can see that we have an increase in antibody concentration that then will slightly go down but be maintained at a lower level. And then if we have a secondary exposure to A, we have a huge spike in the response to A and the antibody titers to A. So in this example, during the secondary exposure to A, we also had an initial exposure to pathogen B, and you can see that there's a slight delay in antibodies to B being formed, but then it's the antibodies to B are formed within subsequent days. So you can see that there was antibody response is not really dependent on the presence of an antigen, it's dependent on the presence of a specific antigen, and that primary responses and secondary responses to pathogens aren't really interdependent. And now let's talk about whether or not memory B cells can gain more affinity to a pathogen after a secondary exposure. So here we're looking at the heavy V regions, the heavy chain and the light chain regions during different types of responses, and we're looking at the CDR1, 2, and 3 regions of both V regions. So again, just as a reminder, the CDR regions of antibodies or T cell receptors are these areas that are hypervariable. So these are the areas that allow for tuning of the affinity to an antigen, and these are the places that are targeted during somatic hypermutation. So in this example, you can see that in the primary response, if you look at the antibodies to this particular antigen, you see that there are some mutations from the germline in the CDR2 and CDR3 regions of both the heavy and light chain. Now, if we look at a similar host antibody response during a secondary infection, you can actually see that we're gaining mutations in both the light chain and the heavy chain, and we're gaining even more during a tertiary reaction, meaning that we're actually gaining antibody affinity throughout each response. So we gain the first level of somatic hypermutation or higher affinity during the primary response, and the affinity grows higher and higher with each response that we gain. So now we've gone through a very broad look at how memory B cells are very unique relative to their naive B cell counterparts, in that they have two different origins that are germinal center dependent and interdependent. So the germinal center dependent responses produce high affinity antibodies. And these memory B cell responses are much more rapid and have a higher affinity for antigens relative to the naive B cell counterparts, which is great because this means that we're launching a highly specific response much sooner, which allows for a faster clearance of the pathogen. And memory B cells can also gain additional affinity to their antigen as they can re-enter germinal centers and undergo subsequent rounds of somatic hypermutation. Now that we've gone through B cells, let's learn about some facts about memory T cells. So if we look at memory T cell responses, today I want to go through where they arise, where the different types of populations of memory B cells, which is predominantly determined by their localization. And I also want to talk about CD4s and CD8s and how they interact in a memory response. So if we look at the origin of T cells, if you were to infect a mouse with a pathogen such as LCMV, which is a virus, you can generate a really large primary CD8 response. And then if you look at the effector cells, it's really curious because you get two separate populations of CD8 cells. You have cells that are very high in IL-7 receptor alpha, and you have cells that are really low in IL-7 receptor alpha. In order to differentiate and to figure out which populations of cells is most critical in forming a memory T cell response, you can take these two different populations of cells and transfer them to another mouse. After transferring these cells to another mouse, you can challenge these mice with the antigen for which they're specific. In this case, it would be LCMV, the virus that you infected the original mouse with. Then what you might expect is that only the mice that have very good memory T cells would respond really well to the antigen and allow for the rapid proliferation of virus-specific T cells. So as you can see here, the IL-7 low cells are represented by the green bar, and the IL-7 high cells are represented by the blue bar. What you can see here is that after challenge, so this is very quickly, a couple of days after challenge, you see a rapid proliferation of the IL-7 receptor alpha high cells, and you see a very low and almost no proliferation of the IL-7 receptor alpha low cells. This means that that memory CD8 cells are actually really high in IL-7 receptor alpha, which is their origin of the memory T cells. In addition, you can see that naive T cells require signals from contact with self-peptides and self-MHC molecules and the cytokines IL-15 and IL-7 for survival, which is what you've learned before. So the interaction between an antigen and a naive T cell, so again, just to remind you, the antigen is presented on an MHC molecule. Most of the activated T cells will actually become effector cells. So these are cells that will directly act to clear the pathogen. Now, upon activation and becoming effector cells, some of the activated effector cells will actually become long-lived memory cells. And as I mentioned in the previous slide, they will start expressing high levels of IL-7 receptor. Then, the cells that become effector cells that express low levels of IL-7 receptor will actually die because they're very short-lived. However, the cells that are long-lived that express IL-7 receptor are long-lived, and the presence of the cytokines IL-7 and 15 will allow them to survive, and then these are the cells that form the memory T cell pool. So this is an example of how two different experiments have shed light on the origins of memory T cells. Specifically, these are those that express high levels of IL-7 receptor after activation. So also, if we look at memory T cells relative to naive T cells, on the left here, we're looking at the naive pool of T cells, and on the right, we're looking at memory pool. And you can see on the left-hand side that the naive pool it increases very quickly right after infection, but then will actually just drop off dramatically. Whereas if you look at your memory pool, we see a high spike during the infection, but this level is maintained for many, many, many days and months and years after infection, which is very critical. So now we've gone through and talked about how memory T cells arise from effector T cells that have expressed increased levels of IL-7 receptor after primary exposure. Now I want to go through the different populations of T cells. So after the formation of memory T cells, we actually get a very heterogeneous pool of different types of memory T cells that have somewhat different functions. These include central memory T cells, effector memory T cells, and tissue-resident memory T cells. Their names should help you remember their functions, but we'll go through them. So, upon seeing an antigen on an MHC class I molecule, A naive T cell will actually differentiate, and we've gone through this in the previous slide. So we have our effector T cells that differentiate, which will then secrete cytokines, express cytokines receptors, and act as fully functional effector cells to aid in the adaptive immune response. The IL-7 receptor alpha high cells will form memory cells directly from some of the effector T cells. Now, as I mentioned before, some of the effector cells will die very, very rapidly after their function is complete. However, some of these effector cells they may become quiescent memory cells, which are very quiet, not really functioning memory cells. However, the memory cells that are derived directly from the effector cells, they will actually form two different effector cells. Two different effector memory cells. So first, they will form central memory cells. So these express CCR7, and they'll stay within the lymphoid tissue. So these guys stay within the lymphoid tissue, as opposed to effector memory T cells, which do not have CCR7, which will leave the lymphoid tissues. So again, our central memory cells will stay within the lymph node, and our effector memory cells will leave the lymph node. So this is somewhat similar, if you can remember, to the origin of our memory B cells in lymph nodes. We had those memory B cells that stayed within the lymph node and many that left the lymph node. We see this too with central and effector memory T cells. The central memory T cells are generated in the lymph node and remain in the lymph node, whereas our effector memory T cells will leave the lymph node and they'll migrate to tissues throughout the body. We also have a different subclass of memory T cells that are called tissue-resident memory T cells. So these are cells that are generated not in the lymph node, like the effector memory T cells, but actually these will remain in the tissue. So we have our activated CD4 and CD8 T cells that will enter the dermis or other peripheral tissues during the primary infection. And their CD69 induction, when they enter the tissue, will actually induce the expression of S1PR1 and maintains the T cells in the dermis. So these cells have left the lymph node during the primary infection, have moved to the site of the infection, and will stay within the tissue where they were effector cells after that response. So I want to make sure that you can distinguish these three cells. So first we have our central memory T cells, which are formed in the lymph node and are found in the lymph node. We have our effector memory cells, which are formed in the lymph node and are found in peripheral circulation and in tissues. And then we have our tissue memory T cells, which formed during the lymph node during the primary infection, but they actually were once they got to the tissue where they were effector cells, got stuck there. So our central memory and effector memories are those that were generated in the lymph node during that primary infection. The central memory guys stay in the lymph node. Effector memory guys move throughout. The tissue-resident memory T cells are the effector cells that were functioning in the tissue and were retained there. Now I want to jump into the interdependence of CD4 and CD8 T cells in their memory. So to figure out how CD4 and CD8 T cells interact on a memory scale, the following experiment was done. So we took, I personally did not, there's other scientists did this, wild-type mice that either had or lacked MHC class II molecules were infected by a bacterium. So again, remember that MHC class II is a molecule that presents antigens or antigen epitopes to CD4 cells. Subsequently, you can see after 7 days of infection, both of these mice, so both the wild-type mice and the mice lacking MHC class II molecules, had an expansion of CD8 T cells that were specific for this bacteria, which means that this initial primary response is not dependent, the CD8 response is not dependent on the activation of CD4 cells. Again, remember that CD4 cell activation is dependent on MHC class II. So without MHC class II, we don't have a CD4 cell response. And you can see that again during the primary response, we don't need a CD4 response to generate a CD8 T cell response. However, after 70 days, both of these mice were challenged again. So this is looking at the memory response. So how well is memory formed between these two mice? And you can see that although the wild type mice were able to also form a memory CD8 response to this pathogen, the mice that do not have CD4 activation, meaning they lack MHC class II, were unable to form a CD8 T cell memory response, meaning that CD4 T cell help is required for CD8 T cell memory. So another way to look at this is in this experiment, memory CD8 T cells were allowed to develop in a mice that was infected with LCMV. Again, this is a virus that's used very commonly in studying memory responses in mice. So mice were infected with LCMV, and memory T cells were allowed to develop. Then, the cells that are specific for this virus were taken from this mouse and transferred either into wild-type mice or into mice that did not have activated CD4 cells. Again, because they lack MHC class II. And what you can see is that in wild-type mice, you can actually see that these memory CD8 T cells are maintained for a really long time in these new mice, meaning that these memory CD8 cells are happy, they're existing, and ready to tackle another bout of LCMV infection. However, in mice that lack CD4 cells, meaning they don't have class II, you can see that there's a plummet of the number of CD8 memory T cells. Again, this is another experiment showing that CD8 T cell memory, which is really critical in cytotoxic T cell memory, is dependent on the presence of CD4 T cells. Now, this wraps up everything that we wanted to cover today with the memory T cell responses. So over the last lecture, we've gone through some general properties of immunological memory, including the fact that immunological memory is mediated by the adaptive immune system, or T and B cells. We've also gone through the fact that secondary immune responses are mainly attributed to memory lymphocytes and how we can use this fact to prevent newborn hemolytic disease. And we also went through the concept of original antigenic sin. When we talked about our memory B cell responses, we talked about their origins. So again, we have germinal center dependent and germinal center independent B cells. And within the context of germinal center dependent B cells, we have those that stay within the lymph node that they originated from. And we also have those that leave the lymph node and circulate throughout the body to other lymph nodes. We also discussed how memory B cells are much more rapid and have a higher affinity for the antigen relative to their naive B cell counterparts. We also talked about how memory B cells can re-enter germinal center and gain higher affinity to their antigen through somatic hypermutation. When we talked about memory T cells, we talked about how memory T cells arise from effector T cells. And so during the formation of effector T cells, there's a small subset that upregulate IL-7 receptor alpha, and these cells become memory cells. And those memory cells that arise can become central memory or effector memory T cells. So these are those that are generated in the lymph node. And again, central memory cells are those that stay in the lymph node, and effector memory cells are those that leave the lymph node and recirculate. And there's also the tissue-resident memory T cells, which will, during the primary response, will go from the lymph node and go to the tissue where they do their effector function. And then they become trapped in that tissue and gain memory abilities such that if there's a pathogen coming in, they're right at the site of infection and can battle that pathogen head on. And then we also discussed how the persistence of CD8 T cell memory is dependent on the presence of a CD4 response. Now, I hope you guys enjoyed this lecture on immunological memory, and that's all we have for today.